Hi, everybody. Thank you so much for attending our Precisionary Scientific Series webinar today. I wanted to welcome everybody, and I'm really pleased to introduce today's speaker, Anna Flury, who is a PhD candidate at the CUNY Graduate Center and is a researcher in the lab of Professor Pinar Ayata at the Neuroscience Initiative within the Advanced Science Research Center. So Anna's research focuses on the role of microglia in Alzheimer's disease, and you'll see this beautiful background that was actually painted by her lab manager and digitized and presented here by Anna herself. And she studies specifically how cellular stress pathways drive maladaptive microglial behavior and then contribute to neurodegeneration. Her work integrates multiple approaches, including ex vivo tissue imaging, ribosome profiling, and both in vitro and in vivo models to better understand how distinct microglial states influence disease progression. And today she will share her work identifying a stressed microglial subset that worsens disease pathology and promotes neuronal damage through the secretion of toxic lipids. And this offers new insight into mechanisms underlying neurodegeneration. So we're very excited to have her with us. Please join me in welcoming Anna Flury. And for those of you attending, please feel free to leave comments and questions in the chat box during the presentation. And at the end, we will have an open Q&A session for any questions. Thank you so much. And with that, Anna, I'm handing it off to you.
Great. Thanks for that wonderful introduction. And yeah, happy to be here talking about my project looking at microglia in Alzheimer's disease. So I'm just going to start with a brief overview of the disease. And yeah, let me know if at any point you can't see my slides. But so most of us are familiar with Alzheimer's disease. It is the most prevalent form of dementia. And you're probably familiar with the outward symptoms. So an overall loss of memory. You might forget where you are in space and time and have trouble remembering appointments, have trouble communicating, tasks that would otherwise be easy for you, like following recipes, navigating traffic, might become difficult. And you might also experience increased bouts of irritation or mood swings. So most of us are familiar with these symptoms because of the prevalence of this dementia. You might have firsthand experience seeing these symptoms in people that you know and love. And there is no definitive cure for Alzheimer's disease. So this is a progressive neurodegenerative disease, and there is no current therapy that will reverse the course or completely stop the course of Alzheimer's disease. And while you might know more about the outward symptoms, you might know a little bit less about what's going on molecularly inside of the brain of an Alzheimer's disease patient. If you're not a neuroscientist, you might not be so familiar with this. But the disease is actually named after Alois Alzheimer, who was a psychiatrist that sort of did the first histopathological characterization of the disease. And what he found were these sort of clumps of proteins that we now have characterized better. And we know that the extracellular clumps that he was looking at are composed of a protein called amyloid, and we call these amyloid plaques. And intracellularly, there are also clumps that accumulate within neurons, and those are composed of a protein called tau, and we call those tau tangles. And there's also now been established a temporal order, a general temporal order that these protein tangles or protein aggregates start to appear widespread in the brain, first being amyloid plaques appearing, and then generally tau tangles come later. And this sort of temporal order has led to the idea that amyloid plaques are an initiating event that lead to a cascade that results in tau tangles and then neurodegeneration and cognitive decline. So a lot of therapies are then aimed at trying to target this initiating event or amyloid plaques. And some of the drugs that do that are actually quite successful at clearing plaques from the brain. But what we know is that if you do that, we still don't get a complete stop or reverse of the disease. So that's led the field to look at other possible sources or driving factors of the disease. And genetic studies have done a lot to try to probe some causal links here, specifically large-scale GWAS studies. And what they found is that there is no one singular mutation in a single gene that will cause late onset Alzheimer's disease, but there are, in fact, multiple risk loci that map to the disease. However, what these risk loci have in common is that they point to microglia. So most of the genes, or a lot of the genes that these risk loci are in, are singularly or most upregulated by microglia. And so briefly, what are microglia? So they are an immune cell in the brain. They are the brain's resident immune cells, actually. And they are very multifaceted cells that do a lot of things to maintain homeostasis in the brain. So as immune cells, they can release soluble factors like pro-inflammatory or anti-inflammatory cytokines, chemokines that signal to the cells around them and can orchestrate an immune response. And they're also macrophages. So they can sense and eat degenerating cells, pathogens, and then they also play an important role in regulating the synaptic landscape. So they can prune synapses, so therefore remove them, or they can also promote the genesis of new synapses. So microglia really do a lot of things physiologically to maintain homeostasis in the brain. And we know that microglia are actually heterogeneous. So single cell studies have really illustrated this point, where there are certain subsets of microglia that can be characterized by a unique gene expression program where they upregulate certain genes compared to other subsets. And in microglia, there is a subset that emerges known as disease-associated microglia, and this was described in 2017 independently by two different groups. This disease-associated microglia subset that has a unique gene expression and emerges in Alzheimer's disease. And this subset is pretty much exclusively found surrounding amyloid plaques. So most of the microglia around these plaques will be disease-associated microglia. And what's interesting about them is that they sort of have purported opposing functions. The two groups that originally described them sort of described them in different manners. So one group purporting a more neuroprotective role, perhaps in containing amyloid plaques, and the other purporting a more neurodegenerative role, actively driving neuronal cell death. And this has since been supported by other studies trying to probe the functional outcomes of DAM, where we see kind of opposite things happening. And so this has led our group to speculate that rather than one DAM subset, there might actually be multiple subsets within DAM, some promoting neurodegeneration and some promoting neuroprotection. And so for the purposes of this talk, I'm going to focus on a DAM subset that could promote neurodegeneration so that we could potentially target this neurodegenerative subset. And I'm going to tell this story in three parts, starting with identifying this potentially neurodegenerative subset, then going into characterizing it, and then delving deeper into the mechanisms at play. So I'm going to start with part one, with the identification. And so to identify the subset, we took some clues from electron microscopy studies. And I'm showing you here such studies that have characterized a subset that is known as dark microglia. So on the left, what you'll see is typical microglia, or what microglial cells usually look like by electron microscopy. And one of the defining features here is, if you look at the yellow outline, this is the nucleus of the cell, and you'll see these patterns of light and dark chromatin. And if we look at dark microglia and the nucleus, we see that this pattern of dark and light chromatin is lost. And also the cytoplasm surrounding cytoplasm is darker. So they're aptly named dark microglia. And the reason that we are interested in these cells in relation to DAM is because, like DAM, they are pretty much exclusively found surrounding amyloid plaques, and they are also emergent in conditions that recapitulate features of Alzheimer's disease. So they're very rare to spot in conditions of health. And the thing about DAM is that they interact extensively with neuronal elements like synapses, dendrites, neuronal dystrophies. So this extensive interaction has given them a purported neurodegenerative role, especially because they're pretty much exclusively seen in contexts of neurodegeneration. And DAM are also characterized, dark microglia are also characterized by cellular stress. So they have aberrant mitochondria and endoplasmic reticulum, giving them sort of suggesting that these cells are undergoing cellular stress. And so we asked the question of whether a cellular stress pathway could characterize a subset of DAM that is neurodegenerative, such as dark microglia. And to try to get at this question, because there's a lot of stress subsets out there, to try to narrow it down, we thought about, well, these microglia are around amyloid plaques, which are essentially aggregated misfolded proteins. So we looked for unfolded protein response, which is a canonical pathway that is activated by misfolded proteins. And there's 3 arms of this response. So we looked for evidence of activation of this pathway in existing microglia data sets for microglia in Alzheimer's disease. And interestingly, what we found was that there was one arm in particular of this subset, the PERK ATF4 arm, that was consistently activated across subsets. And because it was this singular arm, this actually pointed us to another pathway that shares this signaling axis called the integrated stress response. And this is a stress response that can respond to a lot of different stressors, including misfolded proteins, but also oxidative stress and double-stranded RNA. So there's a variety of things that can induce it. Misfolded proteins are one of them. And what happens is that these kinases can sense these different stressors, so PERK, PKR, GCN2 kinases, and then they will phosphorylate a translation initiation factor, which will then facilitate the translation of ATF4, the main transcription factor for this pathway, and which can then facilitate the upregulation of genes in the pathway to restore cellular homeostasis, or if the magnitude of stress is too great, to pivot then to program cell death. But basically, for the purposes of this talk, all you need to know is that we're going to be looking at 3 things to measure ISR activation, which is we're going to be looking at the levels of phospho-EIF2 alpha. We will look at the levels of ATF4 and then the downstream gene expression as proxies to measure ISR activation in a DAM subset. So starting with phospho-EIF2 alpha, so we looked at the levels of this phosphorylated protein within our disease-associated microglia, which we know will be proximal to amyloid plaques. And here what you're looking at is tissue slices from a control mouse in the cortex. And microglia here are in red and nuclei in white. And then, as expected, there's no plaques in our control mouse. And we also then looked at a mouse model that recapitulates amyloid plaques called 5XFAD. And so the plaque here is in blue, and you see the red microglia sort of surrounding it, which is their canonical response. And these will be the disease-associated microglia. And what we see is increased levels of the green phospho-EIF2 alpha in our disease-associated microglia, especially in those microglia that are close to plaques. So that will be our DAM subset. So we see that around 40% of those plaque-associated, disease-associated microglia contain elevated levels of our ISR activation marker. And we also looked at this in dark microglia. And to do that, we used tissue sections using a vibrotome, so coronal sections from our amyloid recapitulating mouse. And we looked for staining for phospho-EIF2 alpha in dark microglia specifically. So the yellow plus here indicates intact endoplasmic reticulum, and the white plus indicates endoplasmic reticulum with phospho-EIF2 alpha staining. So it's sort of, you can see that it's sort of punctate staining here in the endoplasmic reticulum. So we found that the majority of dark microglia did have phospho-EIF2 alpha staining here. So further corroborating for us that cellular stress, in particular the ISR, could be a marker of stressed and potentially neurodegenerative disease-associated microglia. And so this is sort of the model that we were thinking about. And then we also tested this at the level of ATF4. So to do this, we crossed our 5xFAD amyloid mouse with a reporter of ATF4 called Spotlight. And then we could isolate using fluorescence-activated cell sorting our disease-associated microglia based on their gene expression, and then look at levels of ATF4 with using the Spotlight reporter that was crossed into this mouse. And we found was that around 40% of our disease-associated microglia had elevated levels of ATF4. So this was a similar proportion to dark microglia and similar proportion to what we saw before with phospho-EIF2 alpha. And then finally, we looked at the level of ISR gene expression. And so to do that, we did a single cell sequencing of our 5xFAD mouse. And as expected, we could isolate a disease-associated microglia subset from other subsets. And then if we further looked at subsets within disease-associated microglia, we found 3 different subclusters, one of which being characterized by our ISR stressed genes, and the others having cytokine signaling and then A-beta responsive genes. And then we found our stress subcluster at, again, a similar proportion to what we'd seen before, which is around 40%. So at this point, we're pretty confident we had identified a subset of DAM that is characterized by ISR activation. And the next step was to see what the functional outcomes of this subset are, with the hypothesis that this could be a potentially neurodegenerative subset of disease-associated microglia. So now we're going on to the functional characterization. So the first question we asked was if we increase ISR activation, would that expand the stress microglia subset? Would it expand? And conversely, if we inhibit this stress pathway in microglia, would we reduce the amount of stress microglia? And so to manipulate the stress levels in microglia, we use 2 mouse models, which I won't explain in depth here, but if you'd like a more in-depth explanation, feel free to ask me at the end of the talk. But essentially what you need to know is that one of the mouse models, we can activate ISR in microglia specifically, and the other model, we can inhibit ISR in microglia specifically. So they do opposite things. And we can cross those to our 5xFAD or other AD mouse models to manipulate microglial ISR in the context of pathology as well. So we looked at the single cell level to see if we increase ISR, what would happen to our disease-associated microglia subsets. And what we found is that if we increase ISR, we get more of the stress microglia within DAM. And conversely, if we inhibit ISR, we get less of the stress microglia, and we actually get more of the A-beta responsive microglia. So here we sort of hypothesized that this A-beta responsive might actually represent a protective subset that we're getting more of when we inhibit microglial stress. And then we also looked at this in dark microglia. And what we found was that there were a lot more dark microglia when we increased microglial ISR, and then a lot less, almost to control levels, when we inhibited microglial ISR. And so we posited that the ISR might actually be a trigger and it might drive the dark microglia phenotype. So we tested this even in the absence of pathology, and we found the same thing. So if we just gave a control, otherwise healthy mouse, we increased the levels of microglial ISR, we could increase the number of dark microglia. And then opposite, if we inhibit it, then we don't get as many dark microglia. And so now we wanted to use these mouse models to test whether coinciding with these different populations, stress populations of microglia, this could also coincide with levels of pathology. So now this is more of the functional part. So we looked at amyloid, tau, and also synapse loss, which is the biggest correlate of cognitive decline. And so first we started looking at amyloid and amyloid-associated phenotypes in our 5xFAD model with or without manipulating increased levels of microglial ISR. And so what we found is that when we increase ISR, we see a lot less of our microglia, which are here in red again, around our blue amyloid plaques. But if we inhibit ISR, now microglia are going a lot to the plaques. And then we also looked at the plaque load itself, which was also worsened when we increased microglial stress, and neuronal dystrophy, which is here marked in green, was also worsened by increased microglial ISR. And similarly, we saw this with tau pathology as well, that when we increase ISR in a mouse model that can recapitulate tau pathology, we see a lot worse levels of tau. And then conversely, if we inhibit microglial ISR, now tau levels, they go back down. And then finally, we also looked at synapse loss. And here we saw a stronger loss if we added increased microglial ISR, and then we could rescue this back to control levels, actually, when we inhibited microglial ISR. So all of this seemed to indicate that microglial ISR is detrimental to Alzheimer's disease pathologies, and that this ISR-activated microglia subset is potentially that neurodegenerative subset that we are looking for. And because we see this similar pattern where we increase ISR, we see worse pathology. We decrease microglial ISR, we see attenuated pathology. And so now we are going now into the mechanism of how do stressed microglia perpetuate these phenotypes. And so what we thought about here was two things could be going on. So there could be a loss of function, that we get these worsened pathologies because we are losing a protective microglia phenotype. Conversely, it could be a gain of function, that we are worsening these pathologies because we are gaining a new phenotype that is neurodegenerative, or it could be a combination of both those things. But we're focused now on this gain of function, a possible gain of function, because this is something that we could target. So to think about that, we went back to the physiological functions of microglia, and we know that microglia secrete soluble factors that can act as signaling molecules to the cells around them. So we wondered whether our stressed microglia would also secrete soluble factors, and that these soluble factors could be maladaptive. So we moved to an in vitro system to test this idea, where we essentially would just give conditioned media from stressed microglia that we could use a drug to activate ISR, and give the media from that microglia to neurons. So anything that they secreted would be given to neurons, and look at neuronal survival to see whether the stressed microglia are secreting something that is detrimental. And what we found here is that they were. So if the neurons received conditioned media from stressed microglia, their survival was worsened. And then we found a similar pattern actually with oligodendrocytes, which are another cell type, myelin-producing cell type, that is impacted in Alzheimer's disease, and we found a similar pattern. So microglia, our stressed microglia, were secreting something that is detrimental to the cells around them. And the question now we had was, what are they secreting? So to get at that question, we did ribosome profiling from our stressed or stress-inhibited microglia to look for patterns here. And what we found was that, strikingly, in our stressed microglia, there was a striking upregulation of genes involved in lipid synthesis. So all kinds of lipid synthesis in our stressed microglia. And then conversely, in our stress-inhibited microglia, those genes were now downregulated. So we speculated that perhaps the stressed microglia could be secreting, synthesizing and secreting lipids, because astrocytes are known to do that under certain conditions. So we wondered if microglia in this context could also be doing that. So to test this, first we wanted to see whether there was lipids in the conditioned media from our stressed microglia. And so we did lipidomics on the conditioned media, and we found that it indeed was chock full of a lot of lipids. And these lipid species actually coincided with what we found in our lipid synthesis genes by ribosome profiling. So this confirmed that they are secreting lipids. And the next question we had was, are the lipids detrimental? So what we wanted to do was move back to our culture system and then activate ISR in our microglia, and then at the same time remove lipids to see whether it was the lipids that are activated by ISR that are responsible for the reduced neuronal survival. So in order to do that, we went back to our ribosome profiling, and we noticed that this gene called FASN was highly upregulated in conditions of increased ISR. And FASN is actually a rate-limiting enzyme in lipid synthesis. So what we could do was we could get an inhibitor of FASN, and that should greatly reduce lipid synthesis in microglia. So we used this FASN inhibitor. So we activated ISR. We then also co-treated with a FASN inhibitor. So ISR is activated, but lipids are not being produced. And then we gave that conditioned media to neurons. And we saw that now survival is rescued back. So this really confirmed that, okay, it is lipids that are being synthesized by stress microglia that are being secreted and are detrimental to the surrounding cells. And we saw a similar pattern here with oligodendrocytes, so further cementing that. And so now we wanted to move to an in vivo system because we saw all this in vitro. And so what we did was we systematically injected over a period of 3 weeks this FASN inhibitor into our 5xFAD amyloid recapitulating mice. And we saw that we could rescue synapse loss in this way. We also saw this with an ISR inhibitor as well, although I'm not showing you the data here, but we also saw that as well. So now, having seen this in vitro and in vivo, we wanted to know on a more broad level whether we can manipulate ISR and improve on a more broad level memory loss in Alzheimer's disease. So one thing that I mentioned before in my talk was that people with this disease might have trouble with remembering where they are in space and time, and they might have trouble with pattern recognition as well. So what we did was we did a novel object recognition test with mice. And in this paradigm, if you're not familiar, it's just you let a mouse become accustomed to 2 identical objects in an arena, and then you would replace one of the objects with a new one. And if the mouse can distinguish between the object that it's familiar with and a new object, then we say that it has better recognition memory. So it is able to remember what is a new and what is an old object, and it will explore the new object a lot more frequently. But if the mouse has trouble distinguishing this or remembering what is the new object and what is the old object, it will explore both objects at the same level. So there will be no difference. And we can see that here nicely in our control mouse, which is able to discriminate between the 2 objects, in our 5xFAD mouse, which can't do that anymore. But if we inhibit ISR in our 5xFAD mouse, now this recognition returns to control level, so it is able to do it again. And so this is sort of the end of the story here, where we identified our subset of microglia characterized by ISR activation, showed that it worsened AD pathologies, and that this is in part mediated by the secretion of toxic lipids. And with that, I want to thank everybody that contributed to this research, everybody in my lab, all the collaborators from our building and also across the country, and also outside of the country, and all the funding that allowed me to continue with this research in our core facilities, and of course our medicine unit that maintained the mice for us, that allowed us to do this project. And yeah, with that, I will happily take any questions.
Thank you so much, Anna, for a great talk on microglia. I don't have any questions from the audience that is in the chat or also in the private comments section. So with that, I want to say a wonderful thank you to your lab and Dr. Ayata's lab as well. And we hope that we will hear an update on your project with the gain of function studies as well. Thank you so much.
Thank you.
Take care.